Okay, so Chapter 6 is a big one for our class. So the results from this set of experiments is going to be the basis for all of our writing for the rest of the semester. And moreover, you're going to have to be able to do some serious data analysis, interpretation of the results in context, and obviously write the things up in an intelligible way. So fermentation is the process by which any kind of organism, could be single cells, bacteria, and yeast, or multicellular organisms such as our cells, consume energy, get energy out of their stored molecules without the presence of oxygen. So this is an anaerobic process. So in this little podcast, I'm going to cover the two different types of those reactions, and a little bit about the procedure, a little bit about the analysis, and then I'm actually adding a segment about fermentability and why it's relevant to our world and my hobbies. So just so that these images aren't up here without any explanation, of course you're looking at an image of yeast here, one of the commercially important and several commercially important products, including ethanol and bread. And keep in mind that fermentation is important to a lot of our different food supplies. Think of things like yogurt and any host of other things. Okay, so I want to raise this point of fermentability. So not every source of fuel that you give to a cell is going to be appropriate for that kind of cell, whether it's an animal cell or a microbe. It's not going to be appropriate for them to be able to use the energy. And so there's one underlying thing to fermentability, and that is your ability to get to simple sugars. And I've got several inputs on the screen here, and they are grapes or grape juice, barley, which is a grain, and switchgrass, all of which are famous for one reason or another in terms of fermentability. And I just want to give away the punchline here at the beginning. You have to get something sufficiently simple, something sufficiently sugar-like for the cell to be able to metabolize it using fermentation. Okay? And for that, typically you're going to need the right enzyme, and more on that later. But consider these examples first, and what are the net results. If you add yeast to each of these things, so if you add yeast to grape juice, you get wine. There's actually no complications in this, but this makes sense. Grapes are already sweet, they've got lots of sugar. Those can be used directly in fermentation. Barley, on the other hand, if you add yeast to that, what you end up with is bread. And while you get a little leavening, which is actually carbon dioxide being trapped inside the dough as fermentation happens, it's still not on the scale. You don't end up with a consumed liquid. You don't end up with a large amount of the byproducts, which in this case would be ethanol. So there's a problem getting from grain to alcohol, including beer. So, and the real big problem there is all that energy is stored as starch. It's not very sugary. But there's a way to get it to be sugary. And there's a similar problem in our attempts to produce ethanol as fuel from cellulistic materials. So cellulose, remember, is a string of glucose molecules, the same as many other molecules, but it's the structure of it's much harder to metabolize down to single units of sugar. But there are ways of doing that too. And the process would be about the same as converting starch into something fermentable to make an alcoholic beverage, but it's going to be a different set of enzymes. So when you look at the starch molecule, remember that it's a branched set of glucose molecules, each represented by an oval here. And so when you're actually preparing to make beer, which you don't use regular barley, you use malted barley, which means most of the enzymes have already been activated through the malting process to get you relatively simple branch structures like this. And so that when you're in the process of making fermentable liquor for we call it liquor, for making beer. What you do is you put in pretty hot water that's at the correct temperature for two different sets of enzymes to be active. And one of those is alpha amylase, and all it does is clip single glucose molecules off the tips. Oh, I take it back. That's the beta amylase. That's cartooned much better over here. So the beta just clips off single glucoses at the ends, and glucose by itself is obviously very fermentable, as you'll see in the actual chemical equations that'll follow this. And the alpha amylase is actually busted up into smaller and smaller pieces. These give you some not quite fermentable things where the branches still remain, and and then actually smaller molecules like this that are individual glucoses or small strings of not amino acids, but sugars that other enzymes can subsequently cleave into single glucoses, like the beta amylases here. So that's a lot of talking, but the main point is what you need in order to convert a branched complex structure like this into something fermentable like simple glucose molecules is the right enzymes. And in the beer making process, there's a couple important classes called these, alpha and beta amylases. You're not going to be tested on beer making. It seems a bit inappropriate, but do take home that you need enzymes to get to a fermentable material, and you need it to be sufficiently simple for fermentation to occur. And that should be clear when we look at the fermentation. And here they are. So fermentation, you should think of it in two parts. First is the energy capture, and then the second is essentially a cycle that helps renew the different molecules necessary to accept protons so that glycolysis can occur again. And so, as with our photosynthesis, you should be very cognizant of what molecules go in and which come out, and what the net result is. So in the glycolysis step, again, this is the energy producing set. Glucose or other simple sugars go in, a lot of times after being converted directly into glucose. It gets split into two smaller molecules, pyruvic acid, and in that process we've released energy from the glucose molecule and captured it as ATP. Also in that process, we have reduced this coenzyme here, NAD+, to NADH. And in this state, it's now no longer useful for glycolysis to occur again, so we're trapped without a way to make more energy. Importantly, remember that this is in the absence of oxygen. In the presence of oxygen, we're much happier and much more efficient to use the electron transport chain, phosphorylative oxidation, to get many more ATPs per glucose molecule. We do fermentation when we're in a bind and we don't have oxygen. So once we've captured this little bit of energy that we have via glycolysis, we're stuck with pyruvate and a reduced coenzyme, which we need to regenerate back to NAD+. So we're going to go through some oxidative reactions, and we're going to pick our byproducts based on what kind of critter we are. So if you're a yeast, you're going to end up performing ethanolic fermentation, and in that, the ultimate acceptor of the electrons from the NADH is okay, so I'm confused on my redox, but the ultimate product is going to be carbon dioxide, which is spun off, and two different ethanol molecules that go via this intermediate. If you're an animal, you're going to make lactic acid the ultimate byproduct of this. But you notice that in both of these pathways, NADH, which is useless now for glycolysis, is actually recycled back to NAD+. Now that NAD+ can then come back in this glycolysis step and be used to release energy and capture it as ATP. So these are the things that we'll be asking in this laboratory. The biggest one I've referred to is, does the complexity of the substance affect the yeast's ability to ferment that substance? And then we'll test a couple of different things to go with that. Does the nature of the 6-carbon carbohydrate that we put in there matter? What happens with different disaccharides? Are some fermentable and others are not? If so, why not? Same with artificial sweeteners and pyruvate. And this pyruvate question gets at understanding how the cycle of glycolysis and fermentation work. So we need to be able to critically think about why pyruvate would work or would not work. And then we'll address to a lesser extent what the relative rates of those fermentation are on those substrates. So is one substrate preferable? So what we'll do is we will fill up one of these fermentation flasks with a yeast mixture. We will rock it back so that there is no air here. All the gas escapes up here. Then we're going to add our substrate and let it cook for a while. As it rests like this, there's not actually a lot of gas exchange here, so it's essentially an anaerobic environment throughout this whole liquid phase. Eventually what will happen is the gas will collect on the top of the tube, and we'll measure that in millimeters of gas produced. So again, remember, these data are going to be important for the rest of our writing assignments. Collect everything properly, use controls, and then start looking to how you will formulate. In the end, your graph should be nice and neat and serve as a basis for comparing something, comparing the different substrates visually. So you can see, okay, these are all at the same rate. They're clearly labeled for what it was. This one was exceptional. These didn't seem to do much. We can see all these phenomena visually. Note that the time axis is on the bottom, and the independent variable is on the y-axis.